I believe that we're recording. So like I said, this is the rescheduled weekly live online session from last night. So it's the 18th now, the Thursday night, unlike our regular Wednesday nights. And I really do appreciate you joining me. I know that it's a burden having joined yesterday and having to join tonight as well. So what I'm going to try to do tonight is to keep it very condensed, keep it very efficient so we get through the material. And if at any stage you need to go, feel free to do so. If at any stage you have questions, shout out just for me. You can turn on your microphone or you can write a message on the chat. But I am here to help you and make this session worthwhile for you. So like I said, the very first thing for me is to apologize. So what actually happened at the southwest part of the county, we did lose electricity and we did also lose the internet connection. So I was initially debating whether I should ask students to wait around, but I'm glad I didn't. And we simply decided to reschedule this session for tonight, because the power and internet did not get established until late at night. So it would not have been worth of your time to wait around. So without the further ado, here's kind of a quick plan of what I was hoping to do today. So I want to say a couple of words about the exam and lab practical that we completed last week, just to give you an update where we are with those. But really, probably the one that's the most burning question for most of the students is to do with the labs and what is going to happen with the labs. Some students have been able to access this week's labs and some students have not. And fear not, I have a plan B in place and I'll be publishing that really soon. But I first wanted to get a chance to talk with you, whoever shows up on this session. So you hear it firsthand, and we have discussed that through. Of course, in addition to that, I want to spend a little bit of time, and that was where we got last night, about the material relating to the heart. And we really are going to be looking at both of the anatomy and physiology. We look at them a little bit different from what we normally do, where I try to combine them together. Heart really lends itself to address these as a little separate blocks. But my goal is to cover this within an hour or so. And of course, at any stage, if you have questions, just feel free to let me know. So thank you for everyone who submitted the exam and lab practical one. I got that back from almost everyone. And as you saw, we didn't use responders or anything in terms of proctoring. It was a timed exam. And I know that initially the score might throw you off a little bit, that, hey, I felt that I did differently than the score that you got. Remember that the score that you see at this point is the automatic grading that the Canvas does. However, I will go, or I have gone back to go through everyone's exam who have submitted that. I'm just waiting for, I believe, one more exam. And I've gone through your answers multiple times without blinded to the fact who's been answering, and done corrections, given points, even if there was spelling errors, given half points or partial points, even if you kind of slightly missed the right answer, but you provided answer that was relevant regardless. You should be expecting to get the final grades back from me by the end of this week. So what I will be doing, I will be sending out an announcement when the grades for the exam 1 and lab practical 1 have been finalized. So at that time, I would really ask you to go there, have a look through those. What's the grade? What questions you might have missed? If there's feedback, I usually, if you miss anything, I give quite a lot of feedback on those. And if after that you feel that you would like to have a one-on-one session, I'm always happy to facilitate those to go through. If there's something that kind of seemed that you quite didn't understand what was the answer that I was looking for, or if you, for example, have a a concept that you just wanted to go through once more because it didn't quite make sense the first time, so feel free to let me know at that time. I'll post all information about that once I send also the notification that the final grades are ready. And there is a chance if someone feels that the exam or practical didn't quite go your way, I am open to discuss what is the best strategy for us for moving ahead. Should you resit it, or should we look at other options? So fear not, we'll get there together. And this is really also a reminder that I'm also happy to open up any assignments that might have closed, or if you want to reattempt any assignments, not just the exams and practicals, but any assignments that we have had earlier on this course. I would just really encourage you to look if you completed it and you didn't get the score that you wanted, or if you didn't get a chance to get that score, why that might have been the case. Is there something in terms of strategies, how to study or how to approach one of those assignments that we could redefine? I'm happy to also spend some time with the study strategies, learning strategies. I spend quite a lot of time on that, on other contexts for other students. So I have plenty of resources to share on that. If something doesn't seem to work, let's figure out how to make it work. So all you have to do is message me directly. The Canvas messages are not normally the quickest way to get hold of me, but I do answer to emails, everything. So just send me the specific details, which assignment it is that you're looking to get reopened or reattempt, because there's a lot of assignments that we have covered already at this point, and it might take me longer if it's not super clear that which assignment you're looking to retackle. And I normally open those without any problems. So this week, we have a bunch of assignments to complete. So we have the familiar two ones, the LearnSmart and quiz that you've done before. And the third one that's familiar is going to be the practice anatomy, which is basically preparing you for the next practical. Those you know exactly how they work, what's the format, and so on. However, what's new on this week is the two labs that we will be completing, because they are completed in a slightly different way than the labs in the past. And I am aware that some students have had success completing those and some students not so much. And I don't want you to worry if it hasn't worked out for you, because like I said, there is a plan that I have for that, and I will share it with you tonight, and then I'll make a post of that for the rest of the class as well. But let's have a look what's different in these two labs and why they are working for some and not to others. So these labs are something that are known as Phil's labs, and these are built in a little bit of a different way by our book publisher. These labs require Flash Player. So this has, even before the end of last year when support for Flash Player ended, even before that, this was one of those things that was giving students a little bit of headache. So at that time, I created this little help with Phil's labs section on our weekly module. So you will find it under the hard assignments. And there I give details how, at least until the change of the year, until this semester, how most students got the Phil's labs to run. And like I said, that's all to do with this Flash Player that's involved in this. I do realize that Flash Player is no longer supported, so I totally understand that some still don't get it working. So like I said, there's an alternative plan in place. So if you would like to attempt the Phil's labs, the big thing is to make sure that you have A, installed the Flash Player, and B, that you are then enabling it. This means that because Flash has always been a little bit of a not a nightmare, but a little bit of a more of a trickier format to run activities, run labs. Most browsers automatically actually block these Flash requests for activating the Flash player from the websites. So it is very possible that you might need to go and allow it to run before the lab will work. Typically what you would see would be something like what I've screenshotted here from one of my computers where the Flash did not run automatically. So you might see something like a notification that you need to download and install the most recent version of the Flash. I doubt that that notification comes up anymore since the Flash has retired. So that player format is no longer supported. But if you do have a computer that's not brand new, that's not from this year, it is very likely that you still have the Flash player in place and you are still able to access these activities. So what I ask you to do to be able to access those is to look at the very end of the address bar. So this is where you would typically type the address of the website that you're trying to go to if you're trying to open a specific address. So at the end of that bar, pretty much regardless of which player you're using, you will notice that there's some sort of a symbol there. And most of the time, if you have an issue with the Flash player, what you will see is a symbol of a lock, and it might have a red slash running over it. And this indicates that there is some sort of an issue with this. And once you click on that, you can then choose the option which says that please activate the Flash player. And once you do that, that normally solves half of the problem. The second half of the problem is then shown at the other end of this address bar. And it's often shown in a shape of a lock again, or the other symbol that we keep seeing is an exclamation mark. Well, if you see either one of those, you click on that again, and that will open up a drop-down menu. And I believe that I actually have a screenshot of that drop-down menu on the next slide. So it would look something like this. And from there, you will notice there's a section that says Flash, and from this drop-down menu, you simply choose Allow. It is very likely that you might need to do both of these before the Flash starts working. So often it's not just one, it's both of these. So if you do both of these, there is a good chance that the Flash player will start running. And if it runs, you will end up landing on a site that looks a little bit like this, or it does look exactly like this. And this is the Flash program actually running. This is a table of contents of all the fields labs that are available for you. And I would ask you to look for the particular labs, the names of the labs that I'm asking you to do from there. So the names of the labs that we are looking for are going to be abnormal ECG and heart sounds. So you will find them listed in this table of contents, and you then just click on that lab. I think that the abnormal ECG is number 30. I don't remember the other one's number straight off my head, but it is listed there. Once you click on that, what you will see that a page will open where you simply click Next. Then there will be a pre-lab quiz. You do not have to complete the pre-lab quiz if that's something that you're not passionate about. So that's fully optional. So that's not something that I'm expecting you to do. It's good practice, it's fun, but I know that you guys have a lot going on, so you might not have time for that. What I'm asking you to do is that the website pops up this lab exercise as a separate page, and it leaves the old website open, and over there you will see a post-lab, bunch of post-lab questions. These are typically in the Connect in the previous window. So while you're working through the lab, you click answers to this post-lab, And this is how you will be able to complete those questions on that post lab. And this is the element that actually brings the grade to your grade record or grade book here in Canvas. So make sure that you do complete the post lab, not just the lab, but you also get the post lab completed, because that's the element that populates those points to your final grade or your grade for that activity. There is a technical support available from our publisher, McGraw-Hill, in case you get stuck. I do anticipate that since the Flash player is no longer supported, they might not be able to help as much, but that is always a resource that's available there. But like I said, if none of this works, if you are not able to complete these labs, you should not worry. That would not be your fault. That is very possible that that happens. What I'm gonna do over tonight and by tomorrow morning, I will have uploaded 2 alternative versions of these labs. They cover the same material, but in a different way. And you can choose to do either these fields labs or the alternative versions that I provide. And no matter which one you do, you will get the full points for that. I will also be giving you, of course, extra time since we have had issues with these fields labs. So I will put a week from tomorrow for the deadline for those. So you have plenty of time to get those completed. No one has to stay up overnight to get those tackled. I do apologize for this. This is an inconvenience, I'm sure, but this is probably the easiest and quickest way for us to tackle those. So unless you hear anything else from me, this is the plan in place. If there is something else that comes on the way, we will then consider that as well. But that's all that I have to say about the labs. If you have any questions about those, I'm so happy to answer now or later on, so you just let me know. But like I said, there is an alternative way of completing these labs, and you will have all the time that you need to get those done. If there are no questions, just in the interest of time, because like I said, I'm trying to be condensed today, I'm gonna jump on to this week's chapter material. So this week we looked at the chapter 19, which is all about the heart. And how we started is that we had a look of looking back way when we did the basic introductory biology classes. And in those basic introductory biology classes, as well as probably during your BIO201, you might have heard that there's actually 3 different kinds of general types of muscle tissue that exist in the body. And the skeletal muscle is the muscle that we end up seeing on all of those big muscles that generate movement in the body. So it's gonna be those ones that grow when you go to the gym, that really make us show up on our body as muscles. Smooth muscle was the muscle type that we end up finding in 2 places: around various parts of the intestinal tract, so digestive system, and also around the blood vessels. The function of the smooth muscle is to open and close different parts of this digestive tract so that the food flows in there to various places depending on where it needs to go and when it's ready to go. In blood vessels, what smooth muscle does, it controls the diameter of the vessel. And we'll be talking about blood vessels in next week's chapter. So this is just something to put back to the back of your mind, that it is the smooth muscle that then controls how large or how small the diameter of that blood vessel is. So that leaves us with the cardiac muscle. And of course, the cardiac muscle is the muscle type that we're interested in this week, because it is found only in the heart. So that's a very special type of muscle. And just as a review, this is probably familiar to you from the past, but if we look at a single cardiac muscle cell, what we end up finding is that these cells are often branched. So they aren't just a single cell that's a blob, but the ends of these cells have various branches. The other thing that we see with these cardiac muscle cells is that they have these striations, so stripes through these cells. They look a little bit like zebras. You can actually see these striations with a microscope, and they are present there. And in your chapters where you talked about muscular system, I believe in BIO201, you would have discussed what causes these striations as well. The third thing that we end up finding in the cardiac muscle cells is a central nucleus. So usually pretty much in the center of the cardiac muscle cell, you end up finding a nucleus. That stains a little darker. And now we get to the very important part that makes a cardiac muscle cell so successful in its job. So at the end of each branch of the cell, we end up finding something called intercalated disc. These intercalated discs are there to connect this cell to the next cardiac muscle cell. So it is the site where the two connect. And the reason why cardiac muscle cells are connected to each other, and also why they have this special adaptation of the intercalated disc, is that we want, of course, all of the heart muscle contract at the same time. It would not be any use for our heart to have certain parts where the heart muscle contracts at one time and other parts where it contracts at different times. Instead, we want all of this heart tissue to contract at once, and this is what generates the pumping force for the heart. And it is these intercalated discs which then allow for the material and signals to pass from one cardiac muscle cell to another. So this is how these cells are able to function so well synchronized together. And we'll come back to this synchronized nature of the cardiac muscle tissue in a little bit of time again. But at this point, remember that the cardiac muscle cells are specialized in this way. Another thing that we end up finding with the cardiac muscle cells is this ample number of mitochondria. And you might remember mitochondria from when you revised the basic elements of a cell structure. What mitochondria did, they produced the ATP, which was the energy currency in a cellular level. So this is what is considered as the energy that rest of the cell needs to make its functions. Of course, if we think of the cardiac muscle tissue and muscle cells, they function from approximately 2 weeks after the fertilization, and they will keep contracting all the way until you take your very last heartbeat. So I did some math earlier on, and in a day that means that your heart will pump approximately 7,000 liters of blood. This means in a lifetime of an average individual, your heart will contract somewhere around 2.5 billion times. That's a lot. And that's why we need to have a lot of this energy on these cells. And that's why we end up finding a lot of mitochondria on these cardiac muscle cells. What you see here is what I spoke about earlier on, just a zoom in of that intercalated discs disc that connects the two cardiac muscle cells to each other. So in case you were wondering what that symbolizes, that's just zooming into that part that we saw earlier on. So bearing all this in mind, I think the next logical thing would be to discuss where is heart located. And what we're going to do with the location of the heart, we're going to use the anatomical language that we learned in the past. So we're going to use all the correct terminology. And this line that you see here is actually a line where the diaphragm would be located. So you remember diaphragm was this flat, very thin sheet of muscle that separated the thoracic cavity from the abdominal cavity. So heart is located above it. So the term that we use for that is superior to the diaphragm. Heart is located within the chest cavity. So we talk about within the thoracic cavity. If we talk about directional terms again, we have, of course, our rib cage, and within the rib cage, this middle part that was hard was our sternum. So heart is located behind the sternum, so it would be posterior to the sternum. And if we think of the heart's location, well, is there a thing that it would be located in front of? Well, it would be located, for example, in front of the vertebral column. So it would be anterior to that. So there's a lot of these terms that we have already used before when we describe the location of the heart. Another thing that you'll notice is that heart is located fairly centrally within the chest cavity. Often students think that it's all the way to the left. It is slightly to the left if you look at the midline from here. Yes, so it is pointing to the left, but it is much more central than students often end up thinking. And this is why we perform CPR pushing to the middle of the thoracic cavity on the sternum. What is striking about the heart is, though, that the apex of the heart, and apex refers to the sharp tip of the heart. So there's only one place that's a sharp tip. That's apex. And apex of the heart is pointing to the left. And it's also pointing downwards. So it's pointing to the left and inferiorly. And in addition to pointing to the inferiorly, it's also pointing a little bit anteriorly, so in front of us. So that's a combination when we are asked, where is the apex of the heart pointing? So we can imagine it as if it was this fist, and apex would be the pinky finger. So the apex of the heart is pointing to the left, to the inferiorly, and to the anterior. So it's pointing to this direction where it's meeting all of those terms. And I think I normally like to ask in an exam, where is the apex of the heart pointing? So having those in your mind might be helpful. The other thing when we look at the general anatomy, general look of a heart, is that we notice that it looks a little bit like a cone. So heart is a little bit like a cone-shaped structure. And this cone's very tip was that apex that we just talked about. When we look inside that cone, we'll notice that the most prominent part of the heart is going to be these two ventricles. So we have slightly smaller, but much more muscular walled left ventricle. So this is going to be our left ventricle. And then we have this larger but thinner walled right ventricle. And we'll talk about left and right ventricle in a few moments' time. But this is just kind of to give us an idea where the heart is located. And the last locational clue that some students find useful, and we use a lot in the field of medicine, in the field of anatomy and physiology is that we talk about heart's location in relation to the ribs. So your ribs are numbered starting from the most superior going downwards to the inferior direction. So the heart is located between the ribs 2 and 5. And this is important because if we want to get to the heart, for example, we need to put a needle of adrenaline to the heart muscle, we would need to go between the ribs, somewhere between the 2nd and 5th rib. And this space between the ribs, when we discuss, is called intercostal space. So you would use the intercostal space between 2 and 3, intercostal space between 3 and 4, or intercostal space between 4 and 5. So we just had a Valentine's Day. A lot of the time I hear that the way to a man's heart or to a woman's heart is through the stomach, making a good meal for them. As an anatomist, I say it's really between the 2nd to 5th intercostal space. That's the way to get to the heart. So if you want to be writing a romantic Valentine's Day card next year, that's something that you can use. If we look at the heart here, I have now taken a cross-section of the heart that we were looking before, and we're going to be focusing at this point to the fact that heart is located inside this sac. So it's fully enclosed inside a sac-like structure. And this sac is known as pericardium. And this is very important because if you think where the heart is, and we're not showing it on this diagram, but heart would be located between the right and left lungs. Well, that doesn't leave a lot of space for the heart. It's kind of squashed in between everything and then on top of the diaphragm that we saw before. So there would be a danger that heart would be so squashed that it wouldn't have space to move around, which is needed for it to pump the blood. So that's why we put it into a sac. And that sac gives it room to move around. And it also, this sac will have 2 layers. I mean, between these layers, there's going to be a little bit of liquid, which allows these layers to slide past each other. And in that way, facilitate the movement of the heart. And let's look at this, this pericardial sac in a little more detail. What we notice that the pericardium is literally this 2-layered sac. And inside that, we had a pericardial cavity where we had a little bit of fluid. And this facilitated the sliding of these 2 layers past each other. The other thing that I want to highlight from this picture is the actual heart muscle. So the heart wall is made of muscle. And this muscle has different names depending on which part of the muscle wall we're looking. So you'll notice that the outermost layer of the heart is called epicardium. And the term epi is actually something that we use a lot in language in sciences. And I'm not trying to make you a geology student, but I will show you might remember that since the Earth is this kind of a round-shaped structure, most of the earthquakes don't happen right at the surface of the Earth. They happen some way underneath the surface of the Earth. Well, the point right on top of this point where it happens, on the surface of the Earth, is known as epicenter of the earthquake. So you notice this word epi being used again. So epi is something that's right on top or top layer. The most inner layer of the heart then is known as endocardium. So endo, I think of endo is in, sounds a little bit similar. You just change the end to E to I. So it's the innermost layer. And in between these two, you have myocardium, which is the thickest and most muscular part of this muscle wall of the heart. So the order is epi, myo, endo, going from the out to the inside. I could ask it the other way around in an exam. I could ask you to list the layers of the heart wall going from inside to outside, in which case it would be endocardium, myocardium, and epicardium. That would make a really good exam question, I think. Asking the layers of the heart wall, so it would be epicardium, myocardium, and endocardium. That would be the order from outside to the inside, and then, of course, in reverse, if we are going from the inside to outside. Oh, I didn't mean to do that. Let me just erase that. So it would be then the endocardium, myocardium, and epicardium. This is what I meant to do. Okay, knowing now that allows us to move on to think of the first clinical condition that we see. Sometimes when there's an inflammation, or whenever there's an inflammation, we refer to that in medicine with the term itis. So if you have itis of something, it's an inflammation of something. And in case of pericarditis, it's an inflammation of the pericardium. In this case, this structure that we just discussed about, the pericardium, gets inflamed. So there's an inflammation. Well, this would be heard with a stethoscope as this kind of a crackling, creaking sound when you listen to the heart. And we know that that can be a problem. We don't want to have an inflammation anywhere in the body. So pericarditis refers to that. And this would have many problems. This could cause fluid buildup. This could stop the heart from contracting as well, because now it doesn't have all this space within the sac. And so on. The next thing that we're going to look at, we're going to start talking actually about the heart in a second, but I want to put the heart into kind of a bigger picture. Where is heart located and what does it do? So heart is located as a central part of all of the cardiovascular system. Earlier on, we talked about the blood, which was the liquid in it. And next week, we'll talk about the blood vessels, which are the tubing, but it is the heart that pumps this liquid within the tubing. And there's actually going to be two circulatory systems that we see in the heart. But before we talk about those two circulatory systems, I want to bring us back to something that we saw before. We saw that we could have blood that was referred as being deoxygenated blood. Deoxygenated blood had very little oxygen in it. So this blood is usually coming from the various tissues within the body. So it has traveled throughout the entire body and it has given off the oxygen to these tissues. And if you look at the heart, it is really the right side of the heart that carries deoxygenated blood. And then we have, of course, the other kind of blood. And this other kind of blood is going to be oxygenated blood. Oxygenated blood is rich in oxygen. So there's a lot of oxygen in there. And that usually comes from the lungs. And lungs are the place where we add oxygen to the heart. And you'll notice that it is the left side of the heart that contains the oxygenated blood. So there's almost like two hearts pumping as one, if you want to sound romantic saying that, that heart is made of. So there's almost two separate pumps. One on the right-hand side, pumping the deoxygenated blood, which is often shown in blue in diagrams, and one on the left-hand side, pumping oxygenated blood, which is often shown red in diagrams. And if any of the students is colorblind, this is the great time to let me know so I don't waste your time just showing something that looks exactly the same to you. I have had that happening in the past as well. Well, now that we know that heart carries both deoxygenated and oxygenated blood, and that the deoxygenated blood is gonna be on the right-hand side, whereas the oxygenated blood is gonna be on the left-hand side, we are ready to move on. And just before I move on, I just want to remind you, when we talk about right and left, we talk about the patient's right and left. So what looks like left to you, if we have a patient lying in front of you, it's going to be the right side of that patient. So don't let that throw you off. We always think in relation to the patient. Well, heart pumps blood into two different parts of the body. And the first part of the body that we will be looking at is going to be to the lungs. And we'll notice that that's the function of really the right-hand side of the heart. So all of that happens on this side of the heart. You'll notice that blood comes into the heart on the right-hand side, travels down to the bottom of the right-hand side, and then out from the right-hand side. And there it travels to the lungs. All of this blood is gonna be deoxygenated blood. So there is gonna be a little oxygen in it. That's because this blood is coming from here, it's coming from the tissues. Well, now that the blood reaches the lungs, this is the place where we can add more oxygen to the blood. And you'll notice that the blood that's coming back from the lungs is gonna be now rich in oxygen. So it's going to be called oxygenated blood. There's a lot of oxygen in it. And this blood returns to the left-hand side of the heart. So this loop where the blood travels from the heart to the lungs as a deoxygenated blood, then gets oxygenated there and returns back to the heart as reoxygenated blood. This loop is known as pulmonary circuit. So this is the pulmonary circulation of our cardiovascular system. All to do with the lungs. Well, there's going to be another system then you can think of already. You see that I've got a couple of blood vessels that I've left blank there. Let's have a look of those. So in addition to what we saw before, now blood leaves the right-hand side and it has returned to the left-hand side. And now this blood that's on the left-hand side of the heart, it's rich in oxygen. It travels to the bottom of the left-hand side and then shoots out of the heart. And this blood then travels down the aorta to the rest of the body, everywhere else but the lungs. So this is all parts of the body, all tissues. And we call this systemic circulation, because it goes to all systems of the body. Well, this blood, we remember it's rich in oxygen when it goes to the systemic circulation. And here within the tissues of the rest of the body, it gives off the oxygen to those tissues so they can do their job. And the blood that we're left with is gonna be now deoxygenated. So there's very little oxygen left when it comes back from the tissues. And this blood returns then to the heart, to the right-hand side. And we just saw what happens on the right-hand side. So this second loop that we just saw is going to be called systemic circulation or systemic circuit. So two systems that I want us to know. I want us to know the pulmonary circuit, and I want us to know the systemic circuit. Pulmonary circuit was all about taking the the blood to the lungs, whereas systemic circuit is all about taking it to all body tissues except the lungs. Now, if we think of how long of a distance do we have to different parts from the heart? Well, let's think of how far away our lungs gonna be from the heart. Well, they're gonna be right next to the heart. If we think back here where we saw where the heart is located, it was located here in the middle, surrounded by the lungs. So the distance from the hearts to the lungs is not going to be long distance at all. So let's get back to this diagram. So because we see that this distance from the heart to the lungs and back is going to be short, we can tell that the pressure doesn't have to be very high in this system on this pulmonary circuit. This is because if the pressure was very high, it would then end up bursting all these tiny blood vessels here. So we want to have a low blood pressure so we don't break the lungs. The blood doesn't bleed or blood doesn't leak into the lungs. So that's why it's going to be really important for us that the pulmonary circuit, oh, again, I didn't mean to color over that. I apologize. Let me just erase that. That's better. So let's get a highlighter. That's what I asked after. Pulmonary circuit is going to have a low blood pressure. Well, if we think how long of a distance is it from the heart to all other parts of the body, that distance is going to be much, much, much longer. It is a longer way to get from the heart to all other parts than the lungs. So we'll notice that that distance is going to be longer. So for that reason, the systemic circuit has blood that has a high blood pressure because we need to have a lot of force within that blood and within that circulatory system so that it travels to all furthest parts of the body, including your fingertips, your toes, to the very top of your head. That's a long way to go. The other thing that we'll end up noticing here is that there's a lot more blood vessels to cover within the systemic circuit. So we need to pump the blood very hard that we reach all parts of this systemic circuit. So that's the difference between the high and low pressure within the pulmonary system and high pressure within the systemic system. And now if we have a look, I'm sorry, there's a couple of writings from my previous class. I'll just, oh, I can't erase them. Sorry about that. If we look at the heart, and this is zooming into what we saw before. This is a really nice diagram what a heart looks like, but we can look at the heart and see that there's 4 chambers. It's made of 4 spaces. At the very top part of the heart, you'll see 2 spaces, and at the very bottom part of the heart, you'll see 2 spaces. So on the right-hand side, we've got upper and lower space. And on the left-hand side, we have upper and lower space. Well, this upper and lower space has specific terms that we use to describe them. We have atria at the top, so more superiorly, we have atria, and at the bottom, we have ventricles. And this gives us the terminology that we can call the part where the blood enters to the heart as right atrium. I'm going to abbreviate it simply as R8. And then the bottom part of the right side, right ventricle. So I'm going to abbreviate this as RV. On the left-hand side, we have equally left atrium and then left ventricle. So what I have is here a same diagram of the heart that we just saw, but I've just symbolized it with the box as we just discussed. So we have our right atrium, right ventricle, left atrium, and left ventricle. So just what we saw earlier on. I just think that this box diagram is a little bit tidier, a little bit cleaner. Well, we'll see that the blood comes in from the body and tissues of the body to the right atrium. It is going to be within the right atrium where the blood arrives from the body. So this blood is now going to be low in oxygen. It's going to be deoxygenated. Then from the right atrium, this blood continues and travels to the right ventricle. Again, it's going to be still low in oxygen. It's going to be deoxygenated. And from the right ventricle, the blood actually travels out, not through the right atrium. This would be like behind of the right ventricle or right atrium. So imagine that it just jumps from the right ventricle to outside of the heart. And it's going to go to the lungs. And remember, this blood is still deoxygenated. So that's the first part, first circulation part of the heart. Well, now that blood comes back from the lungs, it's going to enter the heart on left atrium. And this left atrium receives blood that's going to be rich in oxygen. So it's going to be oxygenated blood, or you can call it also reoxygenated blood. So it's coming from the lungs. Well, this blood continues from the left atrium to the left ventricle. And again, it's going to be still oxygen rich, so oxygenated blood. And from the left ventricle, it travels out from the heart. Again, it really goes behind the atrium. So you can think of that as a dotted line. And it's going to travel to all parts of the heart through our aorta. And then to all parts of the body. So everywhere else but the lungs is where the blood goes from the left-hand side of the body. So the blood that travels to the lungs is always coming from the right side of the body, and blood that travels to the rest of the body is always going to come from the left-hand side. Blood that's going to go to the lungs is always going to be low in oxygen, so it's going to be deoxygenated, and blood that comes back from the heart and travels to the rest of the body is going to be rich in oxygen, so it's going to be oxygenated or reoxygenated blood. So on that note, we get back to this diagram that we just saw, and we can add the very same structures that we saw before. And what I thought that we can do here, we can follow the path of the blood together through the heart. So we have blood that's low in oxygen, so it's deoxygenated blood coming from the body to our right atrium, and then it leaves the right atrium through a heart valve that we see here. And we'll talk about this heart valve in just a second, but you'll notice it's these flaps here that we see. It has to pass through this structure, and it travels to the right ventricle. From the right ventricle, it travels to the lungs. And again, it has to go through a set of valves located here, and we'll talk about them in a second. So you'll notice that it has to go through these structures, and these are going to be something that we'll be looking in just a moment. Well, just to continue our review, the blood is coming now back from the heart. It's going to be rich in oxygen. So there's going to be a lot of oxygen in it since it's been to the lungs. It travels to the left atrium first, and then from there to the left ventricle. again, it has to pass through this set of valves that we see here. So that's going to be something important that we want to have a look. And again, blood leaves from the ventricle, this time from the left ventricle, and travels up here. And this is going to be our aorta. Sorry, I unmuted myself when I was trying to write there. I'm just going to write it here. And from the aorta, it travels to the rest of the body. So all other parts of the body. And then it returns from the body to where we saw that the blood came to the heart the very first time, which was going to be to the right atrium. So it makes this loop. And now that brings us to have a look of these valves that I mentioned earlier on. So let's take a little bit of time to discuss all of these 4 valves that we saw that control the passage of the blood to from one chamber to another chamber or to the outside of the heart. So let's have a look of them in a second. What I want to do before we jump there, I want to show one more important structure of the internal anatomy of the heart. This is a structure that I tend to ask every now and then in exam. So between the 2 ventricles, we'll notice that there's a wall that separates the ventricles. This is known as interventricular septum. So the ventricular part makes sense. Septum is always a wall of some kind. So what does the inter mean? It means that it's in between the 2. So if you think of the term internet, it tells that internet connects 2 computers to each other. So similarly, interventricular would be between the ventricles. And this time it's going to be a septum that's interventricular septum. Septum just referred to a wall of some kind. So that's all the important parts from the inside of the heart that we need to know, with the exception of the valves that I mentioned that I wanted to take a moment to talk about. So let's have a look at those. So the first valve that I want to discuss with you is gonna be called our right AV valve. That's the one that separates the right atrium from the right ventricle. So for that reason, AV separating atria and separating a ventricle. So it's right AV valve. Another name that we can sometimes see being used about it is gonna be tricuspid valve. Tricuspid valve refers to the fact that the right AV valve has actually, if we look at it, and this is looking at the heart from the top down, it has 3 leaflets, 1, 2, and 3. So it's made of these 3 cusps or 3 leaflets. So just as well as you could call it the right AV valve, you could call it also the tricuspid valve. Both names would be applicable there. Well, let's have a look of that on the left-hand side. Well, on the left-hand side, we have left AV valve, again, separating the left atrium and left ventricle. Can we have any other names for the left AV valve? Oh, yes, we can. We can call it bicuspid valve. And the term bicuspid refers that we have 2 leaflets or 2 cusps that make up this valve. The other name that's sometimes being used is mitral valve. So we can use all these 3 terms meaning the same structure. So on the left-hand side, left AV valve, bicuspid valve, or mitral valve. On the right-hand side, we only had 2 names, right AV valve and tricuspid. Well, we have 2 more valves to look at. Let's have a look of those. So the valve that's separated are right ventricle from the lungs, and before lungs we have a pulmonary trunk. That is going to be known as a pulmonary valve. So it just tells from what structure it separates the heart from. So from the pulmonary trunk, so it's going to be known as pulmonary valve. Same goes for this other valve that we find now on the left side, separating the left ventricle and aorta. That's going to be called aortic valve. And here is where it gets a little bit, I would say, funny at times, is that again, we have a name that we could use for both of these valves. We call them semilunar valves. So the pulmonary valve is known as semilunar valve, and same is aortic valve. So semilunar valve refers to both of these. They're both semilunar. And the term semilunar refers that their cusps are going to be looking like half moon shaped. So they're semi-moons, semilunar valves. So those are all the valves that we need to know about the heart. So if we follow the path of the blood again, blood was coming from the body to the right atrium. It travels through the right AV valve to the right ventricle, and from there to the pulmonary trunk through the pulmonary valve. The blood returns from the lungs, now oxygenated, so rich in oxygen, and it's going to return to the left atrium and travel through left AV valve to the left ventricle, and from left ventricle to the aorta through our aortic valve. So all of the other terms that we could use, just let's add them to this diagram. We had the right AV valve, which could be also called our tricuspid. It could be, and then we had our left AV valve, which was our bicuspid, and it was also our mitral valve. For the aortic and pulmonary valve, we call them semilunar. So those are all of the valves that you need to know. And what we're going to do next, we're going to actually just see the same in this diagram that we drew before. We would have the valves separating the different parts of the heart. Accordingly, our right AV valve, which was also our tricuspid valve, blood coming in and then leaving through our pulmonary valve, going to the lungs. Coming from the lungs and leaving from the left atrium to left ventricle through our left AV valve, also known as mitral valve, or also known as bicuspid valve, and leaving from the left ventricle to the aorta through aortic valve. Okay, sometimes these valves don't work perfectly. So what we saw that these valves were these kind of little leaflets that opened up so that the blood could go one way, but it cannot come back. It's prevented from flowing backwards. So sometimes these valves that we see don't work perfectly. If they don't close properly, so if there's an issue with the closure of these valves, so when they are supposed to prevent blood from flowing, but that job properly, the blood will flow backwards to the direction that we didn't want it to flow. So that is known as an incompetent valve. That valve is not doing its job in a competent manner, so it's going to be incompetent valve. In addition, we can have another thing going on. We can have a valve that doesn't open properly, so blood cannot flow perfectly through this structure. This is known as stenosis of a valve. So these are very stiff, they constrict the flow of the blood, and heart needs to pump much harder for the blood to move through this structure. Well, this would be a problem. This would not be great news for us. And we can fix this problem. Everything can be fixed. So how we would fix it? There's three options. We can make a mechanical valve, which is simply made by man of usually stainless steel, some plastic, and we just put it to replace if the valve doesn't work, and we're good to go. Those are good, but they're not perfect because sometimes they break. Anything that's mechanical can fail. We've seen that. But in addition to that, if we put it to a very small patient, the mechanical valve is not going to grow even though their heart will grow. So we might have to do another surgery, and that would not be great. What we do instead a lot of the time is that we either take that heart valve from an animal. Pig would be a good example of that. Pig heart is approximately the same size as human heart, and we can take their valve and then put it to replace a dysfunctional human valve, and we're good to go. Or we can go to a dead person and take their heart valve and implant it to the patient. So we have those options. Now what we're going to do next to wrap up the talk about the anatomy part, we're just going to look at some of the surface anatomy and especially the blood vessels on the surface of the heart. Just let me check one more thing. So let's have a look at the surface parts of the heart so we can talk about which blood vessels run where. So what I'm going to do, I'm just going to have to move a little bit of windows for myself so I can see better all the parts. So here we are looking at the heart from the front. So it's an anterior view. And we see that this is the right-hand side and this is the left-hand side. We're looking like we opened the patient's chest, and that heart is in roughly at the same location where it should be. So let's have a look what we can tell from that. There is this gap, this depression between the atria and ventricle on both sides of the heart, on left and right side. So this groove is known as coronary sulcus. You remember the term sulcus when we talked about the surface of the brain. These depressions were sulcus or grooves. So we call this depression on the heart that separates the atria and ventricle on the external anatomy, surface anatomy of the heart, as coronary sulcus or atrioventricular groove, because it separates the atria and ventricle. It's the sulcus of heart, so coronary sulcus. Another groove, another sulcus that we see in the heart is separating the two ventricles on the right side and on the left side of the heart. And this is known as interventricular sulcus. And it's going to be anterior interventricular sulcus because it's the one that we see here on the anterior side. We would actually end up seeing a similar depression on the posterior side of the heart. So behind from this view that we see, there would be similar groove separating the two ventricles, and this would be known then as posterior interventricular sulcus. You might think that, well, why do I care about these depressions? Do they really tell me anything? Well, what they tell us, they give us anatomical landmarks for knowing the three coronary vessels that we need to know. So there's three important blood vessels, coronary arteries, that bring blood to the heart muscle itself. So you might ask that, why do we have blood vessels going to the heart? Doesn't it get blood anyways from inside it? It does get blood from inside it, but we have a lot of muscle there. And just like any other tissue in your body, muscle needs constantly oxygen and nutrients. And the outside of the heart is too far away from the blood that would be inside, that the blood would be able to travel through that muscle wall that we just had a look at the very beginning of the chapter. So instead, we need to have blood vessels supplying that heart muscle. So we need to have some blood vessels to do that job. And this is actually the shortest circulatory system in the heart, because, or in the body, because it only covers the heart. And this circulatory system is active only when the heart is relaxed. And we'll actually end up noticing that it is this region where the left ventricle is, where most of the blood vessels goes to. So left ventricle receives most of the blood supply. And this tells us that the left ventricle works hardest of all of the chambers of the heart that we've seen. And here what we see on this diagram is that we have taken a heart away from here, from the bottom. We have our aortic valve here, and then we have our aorta, which then goes to the rest of the body. And we'll talk next week about these blood vessels here on the arch of aorta, but you'll notice that there are two tiny blood vessels here, right after the aortic valve. These are our coronary arteries. So coronary arteries are the ones that we see here that supply the heart, provide the blood supply to the heart. And there's three parts that we need to know. There are going to be actually two coronary arteries that arise from the aorta, but one of them divides into two. So let's start with the left coronary artery. That's often abbreviated as LCA. That arises from here. You remember this was our aorta here. This is where our aortic valve was. So right after the aortic valve, we have on the left-hand side of the heart, and remember this was the patient's left, we have the left coronary artery. Left coronary artery divides into two. It divides on the anterior side. We keep seeing the left, on this side, the anterior interventricular artery. So it's running on the interventricular groove that we just discussed about, and it's running on the anterior side of that. So that's going to be our anterior interventricular artery. Sometimes it's in medicine described as left anterior descending. Both would work there. The other branch that the left coronary artery divides into is known as circumflex artery. And this circumflex artery goes around the heart to the backside. So we would not see it. It would be dotted line, or it would be hidden behind there. But it's known as circumflex artery because it circles the heart. It goes behind it. So often abbreviated as CX or LCX. It goes to the backside of the heart. So it travels within this coronary sulcus that we saw, atrioventricular groove that we saw between the atria and ventricle here. We have our atria and ventricle there. Well, let's have a look of the right coronary artery. Right coronary artery arises opposite to the left coronary artery. So here we have our aortic valve, our aorta, which then goes to the rest of the body. And on the right-hand side, we have the right coronary artery coming up. And you'll notice that right coronary artery travels again on this atrioventricular groove between atria and ventricle. And it has various branches that come out, but you don't need to know about those branches as long as you know that the right coronary artery travels there in this space that we saw in the atrioventricular groove, or also known as coronary sulcus. And it travels in there and wraps around the heart and goes again to the backside like we saw with the circumflex artery. So it would travel also to the back of the heart. So those are the coronary arteries that we need to know. And in complementary systems, arteries, we of course have veins. So there's some major veins that you can notice from the heart and coronary circulation. And what we're going to do now, I'm just going to take literally 2-minute break, and we will then continue by talking a little bit about the physiology aspect of the heart. So we should be back. And what we're going to do now, we're going to continue talking about the physiology of the heart. So we've seen all the anatomy, and now we're going to move on to discuss the physiology. And you might remember that when we talked about the nervous system in finishing of the BIO201, we saw that the action potentials traveled within the nervous system, within the neuron, and they had a shape in terms of when we followed the membrane potential in one single part of the nerve cell that looks something like this. We had the upshoot, the plateau, downshoot, undershoot, which was the recovery time, and then back to normal. And it was somewhere around 70 millivolts. And it reached the 0 millivolts and became slightly positive at the top. Well, just like we had action potentials within the nerve cell, we have action potentials within the heart. The big difference here is that whereas these nerve cell action potentials were very, very quick, they propagated within 2 to 5 milliseconds, these action potentials are much slower in the heart. So cardiac action potentials take somewhere between 2 and 400 milliseconds. You might wonder that, well, that doesn't really make sense. Why is it so much slower? Well, the explanation for that is that if you think of what the heart needs to do, heart needs to contract so that we can squeeze the blood first out of the atria to the ventricle, and then ventricle contract so that we squeeze the blood either to the pulmonary circulation or to the systemic circulation. Well, the blood doesn't move as fast as a nerve impulse, so we need to give it time to make its journey from one chamber to another chamber. And what I have here, I have the full explanation of each step of this action potential within the heart muscle and heart electrical conduction system. But what I really want to highlight is the fact that it is much slower than the nerve cell action potential. And on that note, we're going to follow the journey of the electrical currents through the heart, adding this physiology to the anatomy that we saw before. You remember that heart was this kind of a 4-chambered structure. We've seen that before. Well, let's now take the anatomy that we know already and combine that with the physiology. So everything starts from this point here that you see. And this point that we see here is really what is the pacemaker of the heart that controls the cardiac beat. And it is, of course, known as a SA node. And the SA node stands for sinoatrial node. So that's where the heart rate is initiated from. And once it has been initiated on this sinoatrial node, which is located on the superior part of the wall of the right atrium, it travels and covers both atria, both on the right-hand side and on the left-hand side. Remember, the atria were the upper part. So it travels the walls of both right atria and left atria. And what it does, in this way, it sends the signal to the cardiac muscle on these atria to constrict, to contract, so that they squeeze all the blood out of those, and the blood travels, of course, to the ventricles downwards. So now we have had the electrical current started by sinoatrial node, and it has traveled through the atrial wall. Well, after this, remember the blood takes a while to travel, so we reach a point that is known as AV node. Well, AV node is located between atria and ventricle on the right-hand side of the heart. So between the right atria and right ventricle. This AV node is a delay station where we hold the signal for a second before we let it pass further. So once we have hold the signal for a little moment, that has given time for the atria to contract. And after that, the signal keeps traveling down the wall of the heart, first within the bundle branches, and then down the Purkinje fibers, these are traveling within the interventricular septum. So the structure that I talked to you about when we were looking at the inside parts of the heart. So that's why it was an important structure. And eventually from there, once it's reached the apex of the heart, it travels up the ventricular wall. And while it travels up the ventricular wall, that then triggers the contraction of the ventricles. So now blood passes from the ventricles both to the pulmonary trunk and to the aorta. So blood is squeezed out. So this is how the electrical currents travel through the heart. And by this slower propagation of these currents, we're able to ensure that the blood has time to travel from one chamber to another chamber and then out of the heart. And sometimes this conduction system doesn't work the way we would like it to work. Sometimes there's failures in it. And let's have a look at a couple of examples of that. I think the pathology is always something that students enjoy. If we have irregular heart rhythm, so this contraction doesn't go like it should go, first atria contract, then ventricle contract, atria, ventricle, atria, ventricle, atria, ventricle. If this doesn't happen, if we have irregular contraction, that it really doesn't go in synchrony, we talk about having arrhythmias. If we have a particular type of irregular heart rhythm where these contractions are especially rapid, so they go all over the place all the time, there's no synchronization whatsoever, we talk about the heart having gone into fibrillation. So both of these would be problems for us. And the tool that we use to deal with this sort of a problem is known as AED. And that stands for Automated External Defibrillator. So automated tells that it's going to work on its own. External, it's not in your body, it's outside your body. And defibrillator tells that it's going to give you an electric shock that then hopes to stop that fibrillation from happening. So really CPR is important. CPR is a great skill to have, but rarely does anyone just jump up from having needed CPR and then live their life happily. Usually it is the AED that saves lives, giving that electric shock that then realigns this cardiac rhythm. So I really strongly encourage you, if you have a chance, find out where the AED is in your school, in your campus, in your place of work. And there are great courses where you learn to use it, but don't be afraid. That machine tells you step by step what you need to do if you ever need to use it. It tells you exactly where to put the pads. It tells you what buttons to push. It tells you to stand clear of the patient. It tells that it's going to give an electric shock to the patient. And after that, it tells it's safe to touch the patient. And the goal is that the electric shock, we hope that that brings the heart beating, these heart conduction contractions back to what they should be in a patient. Well, that was our external defibrillator. Some people have problems with this cardiac rhythm constantly, more regularly than just having a need to do that occasionally. They might have an implanted pacemaker. Implanted pacemaker is a small electronic device that we place within the chest. And whenever there is an issue with irregular heart rate, this pacemaker gives a shock to the heart and returns the heart to its normal beating rhythm. So you might have seen or heard of someone who's got a pacemaker. It shows up just a little underneath the skin. An interesting thing that students find is really, it is just a little box from which we have a metal wire that wraps around the heart. And that box gives an electric shock to that metal wire that then shocks the heart. When we have these patients coming, this is not something that's in your exam, but when we have them coming after they're dead, we do remove the pacemakers from them. They need to be removed before these patients can go either cremation or burial. And actually, pacemakers do get reused. So they get fixed after they've been taken away and then reused on new patients. So what we can do, we saw all this stuff about these electrical currents traveling within the heart. What we can do, we can actually measure them. And how we measure them is with the help of something known as ECG or electrocardiogram. So what electrocardiogram does, it's a trace that we see as a result of putting various LEDs that measure the electrical current within the body. And how it measures the electrical current is that it actually isn't looking just one of those spots that we had a look of. It sums all of those electrical currents together, and then we look, what do they look like? But because the LEDs are positioned in different parts of the body or in different parts of the chest, we are able to get more information than just the sum of all of it. We're able to tell that in one part, the trace look different than in another part. And that gives us information both of the anatomy and physiology of the heart. So let's have a look of a typical ECG trace. What it would look like and identify few important elements from it. I'm not expecting you to become experts of analyzing ECG traces, but I think that it would be good to have just a basic appreciation that there is certain things that we need to notice from those traces. We wanna understand the basic elements. So here we have a very simplified electrocardiogram. So very simplified trace that we might see. And you'll notice that on that trace, we have certain bumps and certain parts where the trace shoots up and down and up again. And each of these bumps or movements has a name. And these names are going to be important as we start analyzing these traces. So let's have a look and give name to these different elements. And we're going to start with this first bump that we see on this trace. This is known as a P wave. This P wave happens when there's depolarization of the SA node and atria. So that's when the SA node and atria depolarize. So that's our P wave, the first element that we see. The second element that we're gonna see is gonna be actually a combination of three parts. It's gonna be called our QRS complex. And what we see when we see QRS complex, it tells us that our ventricles have now depolarized and our atria repolarizes. And the final part of the ECG that we see is this bump known as our T wave. And T wave tells us that now ventricles repolarize. So really looking at all of these different bumps and events tells us what the heart is doing at that very moment. And what's important is not just identifying these different elements and bumps and depressions and increases, is also looking at distances between them. And when we analyze ECG, as you will do in your lab, if you get it to run, if you don't get it to run, I have another way of doing that. I have a lot of ECGs for you to have a look, even in a paper version. What we do, we look, can we see each of these particular events? And if we can see it, are the distances between these events what we would expect them to be? Is it what we expect? And now to kind of wrap up or summarize some of the mechanical events that we need to know about the heart, there's two terms that are going to be really important for us to know. During systole, the heart is contracted, or the particular part that we talk about is contracted. And of course, what that means that, for example, during systole, a particular chamber is constricting, it's becoming as small as it can become. It's shrinking in size. During the constriction, so during systole, this chamber is pushing out all the blood from it. It's getting smaller and smaller, and as it's becoming smaller, it squeezes all the blood out. The other term that I want to show you is gonna be our diastole. What happens in diastole is the opposite of that. In diastole, whether it's the heart or a particular part of the heart, particular chamber, now that chamber is relaxed. It grows in size as big as it can. So it dilates. That's how I remember it. And when it dilates, blood can flow inside it. So in this case, blood is coming into that part. So those are the two terms that are going to be especially important for us. A single cardiac cycle refers to the one full complete heartbeat. So when we listen hard with a stethoscope, what we end up hearing is this kind of a lub-dub sound. So there's two sounds that we hear within each cardiac cycle. And this lub-dub sound is actually telling us what happens at different parts of when we hear those sounds. This sound is made by the heart valves, and when they shut close, that's when you hear that sound. So the first sound that we end up hearing is gonna be a little bit like that lub, if you wish to describe it. This sound is generated when the AV valves close. So remember when we talked about the AV valves, these separated the atrium ventricle on both sides of the heart. So on the right and left side, when they close, you hear the first heart sound. When we hear the second heart sound, which sounds a little bit more like dub, that's a result of semilunar valves closing. So either our pulmonary or aortic valves, or both of them. That's the second heart sound. And we'll notice that there's a small pause in between the next set of lub-dub, lub-dub, lub-dub. That pause is the time during which the heart is relaxed. So we are in diastole during that period. So these sounds, when we listen them with stethoscope, we can hear a lot about how the heart works. We can learn a lot about anatomy and physiology of the heart without even ever touching it, going inside, just by listening it with the stethoscope. So that's pretty cool. And there's different parts within the chest where the doctor will place the stethoscope to hear about each of the different valves, how it works. So when you go closer to that valve and further away from the other valves, you will hear especially how that particular valve is working. So that's why the doctor will probably move the stethoscope a little bit on your chest to listen what noises are or what sounds are being heard. Sometimes when there's issues that, for example, the valves don't open properly, you remember we talked about incompetent valves and we talked about stenotic valves. If there's issues with either opening or closing of the valves, we will hear something that's described as a heart murmur. Heart murmur is this sound when the that we can hear with stethoscope. It's kind of a high-pitched or sound or crickling sound or swishing sound, depending on what's happening. But these are known as our heart murmurs. So heart murmurs are what we can hear with the stethoscope if we have issues with the blood not flowing where it should. There's some sort of an obstruction. Yes, ask me a question what you had. Heart quiz number 23. Let's jump there. I'm gonna just, if you give me 2 seconds or 2 minutes, we're gonna wrap up this chapter and then we're gonna go to the heart quiz question number 23, if that's okay. 2 minutes and we're done. Okay, so last thing that I wanted to mention about the heart is that when we look at the amount of blood that's pumped by the heart, that's actually a quite interesting topic in a sense that typically your heart puts out about I'm going to take a pen, something like 5 1.25 liters in a single minute. But this amount is, if you don't use liters, if you talk about gallons, about 1.5 gallons. But this amount can get high when the heart is working especially hard. What happens in this case, it can come up to 20 or even 25 liters per minute. So it can become more than 10, almost five-fold in the amount of blood that gets pumped out. Well, how do we know how much blood is being pumped out? Well, the term that we use to describe the amount of blood being pumped out is known as cardiac output. That's abbreviated as CO. And cardiac output is measured as liters per minute or milliliters per minute. And how we can calculate the cardiac output is that we look how quick your heart beats. So what is your heart rate? How many beats per minute you have? And that's what the doctor or the nurse will be doing when they take your heart rate and measuring the pulse. This number of heartbeats per minute gets multiplied by the amount of blood pumped out in a single pump. So by our stroke volume. So single contraction, how much blood gets squeezed out. And when you multiply these two, heart rate and stroke volume, you get the cardiac output. And that really is all that I had to say about reviewing this chapter. So I would have reminded you that make sure that you use tutoring services if that helps you. I'm available also for one-on-one or group meetings. And now I was going to say, let's do questions. So let's now jump to the hard quiz question number 23. That's something that we had a question about. Let me just jump out of this. And what I'm going to do, I'm going to open you a view of my screen. You should be able to see my screen here. And we're going to go to the hard quiz. And I think the question that you had, if I remember correctly, was question number 23. So
yeah, let's 23.
Okay, let's have a look of that. That what's there that's I don't remember exactly the question, so it's gonna be helpful for me to as well remind me what the question was. So 23. Was it this question, the events of the cardiac cycle? Let's have a look of it together. So what you're being asked here is to put the cards or the pictures into proper parts of the cardiac cycle. And these are terms that we have not completely covered, but we've kind of touched upon. So when your ventricles are filling, you'll notice that the blood will be flowing into these lower chambers, to these more inferior chambers. So that's why we would be putting these two cards there. So you will look at wherever we see an arrow going to the two lower chambers. So to the right ventricle and left ventricle. We see it here and we see it here. So we would put those cards there. When it's isovolumetric contraction, what isovolumetric contraction refers to is when your heart valves, the AV valves, are closed, so blood can no longer flow back to the atria. It's stuck within the ventricle, but the pulmonary valve and the aortic valve have not yet opened. So the heart ventricles are contracting, but the blood is not escaping from the ventricles yet. All of the valves are closed. So the pressure inside the ventricles is growing. So that's our isovolumetric contraction. So in that picture, we want to see that all of the blood is inside the ventricles, and there's force being pushed against these valves, against these AV valves. But the blood is not yet leaking out from the pulmonary valve or aortic valve. The isovolumetric relaxation refers now when these valves have opened from the ventricles and the blood is returning to the atria. So blood is now coming to the atria, the AV valves are closed. So blood builds up within the atria. That's isovolumetric. Again, the pressure is building up inside the atria, both on the right atria and left atria. And finally, the ventricular ejection is the part that would take place when the ventricles contract, and that forces the pulmonary valve and aortic valve to open. So the blood flows out of those. So in this, we would see some blood still in the ventricles, but blood is mainly in the pulmonary trunk and aorta. Does that help a lot at all? That's a kind of a tricky question because I would have placed the ventricular ejection here between the isovolumetric contraction and isovolumetric relaxation.
Yeah, it helped a lot, Professor.
Excellent. Is there any other questions that you're kind of working and you're wondering that, I wonder what they're meaning with this?
Yeah, I have a question. It's not like on what we're working on, but it's on my graded endocrine quiz, I think.
Okay,
I sent you a message for the questions I missed, but you didn't reply to me on time. So you gave me full credits, but later you like dropped the credit after that. So I don't know.
No, let's have a look. Absolutely. So I'm going to pause the record. So I just turned on the recording again because this is probably a question that few students are going to have. When you are working on the labs, how are you able to access those? So if you would be willing to share with me your screen, what you do, you look at the Cisco WebEx meeting window that has opened to you, and there should be a button that says share. It looks like this little box that has an arrow coming out of it.
Okay, I think I see it now.
If you click that, and when you click it, you should be able to see something like a screen 1 or what options does it give to you?
It gives me a screen, I think.
That's perfect.
You see this one now?
That's it. Now, if you click on that, now I'm able to see what's on your computer, so we can try to work on that together. So
yeah, so when I opened it like this, I launched it, then it showed me this. I did this already.
Yeah, let's
then I did continue. Then this.
Okay. What I would ask you to do is push the arrow on where you go back to the previous page on the left upper corner. There's an arrow pointing to the left side. So let's go back one step.
Go back? I don't think we can go back. We can only go forward.
If you go to the left top corner of the screen, after the address bar, there's an arrow pointing to the left-hand side.
This one?
You can click that. That will work too. Perfect.
Oh.
And then I think if you click the next, that should open it.
Oh, okay.
And what you'll see that there is the previous page, the question 1, abnormal ECG lab, which is the post-lab questions, there available on the previous site. So just click on that as an X. And you can just skip the quiz. You don't have to do the pre-lab quiz at all. So you can click that. And that brings you to the lab exercise. And as you're working through the lab exercise, you're getting the answers for the post-lab quiz. So you'll notice that the post-lab quiz is open in the previous tab that opened before. That's one on the left at the very top that you see. So this is the lab that I'm asking you to complete. So basically what you're doing here, you're doing everything that we just discussed about how ECG works. You're measuring it from a patient, and then you're getting traces and you're analyzing. And it will tell you step by step what to do. So what you want to be doing, you want to connect the right LEDs to the right places. So you literally click away based on what it asks you to do.
Like the chord or like another one?
Let's have a look together.
Do I need to first?
No. Okay, so what you want to do, click on the blue plug. What can you do with that? If you click on that, perfect.
I did that.
Well, are you able to drag it to the number 1 on the data acquisition unit?
This one? I think
Yes.
so.
If you drag it there, does it go?
Yeah, I did that.
And then you want to put now the LEDs on the A. So you take the red electrode and drag it into Amy's right wrist. And remember, right is Amy's side, not your right, Amy's right. So click on that, keep it down, and drag it.
That's this side, right?
Wrist, yeah.
Or wrist. Down here?
Down there. If you release, we didn't quite go. Let's try again. That seems good. Perfect. Then you want to take the black electrode and attach it to the Amy's left wrist. So on the other side, just like what you did before. Perfect. And finally, we want to attach the green electrode to Amy's left ankle. So you click on that and drag it to the ankle and left. Remember, ankle is all the way down just before the foot.
Oh, yeah, that's true. Sorry.
That's it. No. Perfect. Now you are going to push the start button to display Amy's EGT on the screen. So start button is at the upper right corner, little up, on that little screen that you have there, little higher up, at the very
higher?
Not the start button.
Not the on button? Oh, the start button.
Yes, start button. That's what you want to click. Little down. That's it. Perfect. And what you're seeing now, you're seeing Amy's EGG. So you can actually go through it and look back and forth if you're wanting to see what's going on. So I think you're seeing what you're wanting to see. And if you want to stop now the recording of the Amy. Perfect. And what you can do, you can actually move the time on that screen. If you go to that little dot underneath the heading time, that's dark.
Mm-hmm.
And you drag that back and forth, you can see the ECG trace at different times.
Like the same.
Yeah, they look the same. It's regular. There's nothing unusual. It's looking good. So you can also click the journal that's on the screen and write any notes that you might want to wish to make. So you can, for example, write that it looks regular. And then you can just close the journal. You have made the notes that you needed to do. So you do the X. You can also print Amy's ECG trace by pushing the, I believe it's the P button next to the start. That. And then you can write your, if you wanted to print it. You don't have to print it if you don't.
Okay.
Yeah. And you can click that. You can close. You can also fill more information. You have now collected all the information that you needed, and you can click on the post-lab quiz. Just perfect. So what did Amy's ECG trace show?
No abnormality?
Right. Sorry, what was the question?
No abnormality? Answer, I mean.
That sounds, yeah, that sounds good to me. Submits. Good job. Then you go to the next question. Which way?
Oh, we didn't Brian TKG.
Oh, we might need to go back to the lab exercise. Let's click back to the lab exercise. Okay.
It's telling me not available. It's not available anymore.
We will get it available. So we should have probably done more patience. So click on the X on that. And if you press the X on the Phil's website, on the tab, just the tab. Go a little.
This one?
Yes, that one.
That one?
Yes. And now you click again the abnormal ECG. So let's see if we can get the other dude done. So you can just click away, next, and skip the quiz. And we do the same things we did before. So you first connect the blue to the recording input one. Great job. And then you connect the red to the right wrist. It's a little bit temperamental that it has to be perfectly there. And then black to the left wrist and green to the left ankle. Perfect. And then you click start to collect the trace. And then you can stop that. And then you can scroll through that like we did before. Perfect. And you can write there that it looks normal to the journal. So Amy, normal ECG trace. Perfect. And it also asked you to make print. If you click on that print, you don't have to fully print it, but you can press the P and write your name to there. That you can put your name. Perfect. And print tracing. You can save it as a PDF. If you click the destination.
I can actually print it, actually.
Okay, well, you're good. Excellent. So then you're all set. Then how do you get to the next volunteer? Let's see.
Oh, yeah.
You click on the name Amy. That's on there, on the next to her. And you should get another volunteer. Anything that you looked at? Nothing.
Nothing popped up. No.
Oh, it's on the screen, on the screen next to you where the trace was, where there's a drop-down menu.
Oh, yeah, I saw it now.
No,
Yeah.
it took me. Let's click on Brian. And let's record his ECG. So start. What do we see there? There's something weird going on, right?
Something.
So you can push stop. and we can make some notes to the journal. So we see the initial P wave, that small jump before everything else starts, right?
Mm-hmm.
But if we get to the QRS, what we're noticing that the QRS complex, it's not working the way it should be, right?
Yeah, it doesn't look right.
It looks like there's almost 2 R's. R is almost like twice, and then it shoots down. So that would be a good observation to write about Brian. So yeah.
So we should write
that Ryan has Ryan's QRS complex. Yeah, I know it's hard to get the QRS complex has 2 peaks for R.
2 peaks on R?
Yeah, on R for Roger. Perfect. That's a good observation. So you can close that. And then you can do the next page.
Next one.
Right? Yeah.
Increase
Perfect.
R.
What are we seeing there? There's then something odd happening.
Yeah, I think the QRs, the Q is increasing and decreasing.
Yes, that's absolutely, you're correct. There's something weird. So if you stop that recording again, and then we can see, like you said, that the QRS grows super high. What was his name? I don't remember.
I think Chris.
Yeah, Chris. Perfect. And I think that it's every third or so QRS complex is elevated. Or looks high. You can write it however you wish. Perfect.
Sequence, I think.
Yes, that's a good term to use there. You are doing great notes. So if you click that, perfect. And we do the next patient, which was name was
Debbie.
Yeah, let's do her. So start again the trace. And let's see what we're seeing there. Oh my goodness, there's something concerning going on, right?
Yeah. Yeah.
If you
the ST segment, I think.
Yeah, there's something really weird. If we look at, it looks almost like we're not seeing P at all, or P is like multiple times, right? And then, like you said, the T seems really big as well. So you can make a note of that. Perfect. Excellent. Perfect. Those are really observations. Now you can. And
I think that's it.
That's it. So if you click on the question 1 on the previous tab, yeah, you can do that. So let's do Amy's. There were no abnormalities. Brian's. What did we have?
QRS. QRS complex.
Yes, that's correct. There was an issue with the QRS. Perfect, good job. And then we look at how does it differ from Amy's? The QRS complex had, what did you notice that there was going on?
I think it had smaller amplitude, I think.
I think
Was it quicker?
It had
two picks.
You remember we saw 2 R's. The R came up twice. You remember that? So let's try that.
So 2 picks?
Yes.
Yeah.
Good job. And then how does an action potential travel from Brian's AV node to the 2 ventricles? So you remember we discussed this to the 2 ventricles. What do we have? True AV bundle, branches, Purkinje's. Which one do you think that would be?
I think it was A, through the AV bundle, the bundle
Yes.
branches are...
That is correct. Perfect. How could a double peak in the QRS complex be produced in Brian's ECG?
I think the AV node produced 2 action potential.
You can pick that. That sounds good. You can do that.
No.
So what we wanted to look at there is that there's 2 bundle branches that have slightly different conduction velocities. So we're seeing that twice. But since we're working together, I'll later on correct that for you. Don't worry about that. You can continue. And Chris's ECG trace, what did we see there?
I think we see the P. No. No. I think the QR. I can't remember. Let me go back. Can we go back? No, I don't.
I think that what you see is that you have the 1 abnormal followed. Hold on.
I think there's 1 abnormal followed by 2 normal.
Yes, that sounds correct. Every 3rd one was abnormal one. Yes. Good job. And Chris's ECG, which wave was not present?
I think it was the QR. No.
I think that we saw a high QRS.
Yeah, I think it was a Q. Wasn't present. 2 waves, maybe.
You can guess that. Don't worry, we'll work through it together then at the end.
The waves, the 2 waves. I can't remember. Let me see if I can. Okay, we can go to the journal. That's true.
Yeah.
I think it was a QR.
Okay, you can pick that.
Doesn't sound right.
Let's have a look.
Yeah. P.
So what we saw was the one missing, but we probably should have written a little more notes. So I'll take a little bit of that blame. Let's continue. If no P wave was present in Chris's ECG, the atria were not contracting. Source of the rhythm for the ventricles in the abnormal ECG tracing.
I think the essay node. Maybe?
Okay, that sounds good. Remember, that was where everything started. So it would be the conduction from the AV node onwards. Remember, SA node was the pacemaker where everything started. You can click continue. Which waves can you see clearly in depth ECG tracing that are at intervals similar to those seen with Amy's?
I think it was a P wave and T waves. Because she had an issue with a T wave.
Yeah, she had issues there. The T wave was large and P wave was everywhere. But we did see, though, the QRS, didn't we?
Yeah, we saw the QRS.
Yeah.
So I think it should be C. B, I mean.
Yeah, that sounds good to me. I would have picked that. Good job. And one more. It's way.
No.
Which of those
I think she had multiple P waves.
I would
Because it was
agree with
Deb.
that. Yeah,
But I don't know if it was indicates in the actual. I don't know which one.
Let's have
P
a
or
look
D.
let's have a look what we remember about P waves. That where did we see the P waves? You remember we talked that referred to the depolarization of the SA node and atria. So would it be, which one would you guess? So P wave tells us when the SA
Atria, I think.
Atria. Yeah, that's what I would have picked. Good job.
Yeah, so I can retake the quiz.
So yeah, you can do the restart, start the quiz if you want to do it again to get everything correctly. Or you can just go to the previous tab where it says question 1, abnormal ECG.
Question 1.
You can click next. What happens if you click next? Perfect. So you got that. What you can do if you want full points, you can click on restart the quiz and
yeah, I can do that.
Yes.
I think that's what I'm gonna do.
Perfect. And then you notice that on the previous tab, you had the question 1 abnormal ECG left open before the lab opened. If you go all the way to the top of the screen.
This one?
I can't see your, go higher up, higher up, higher up. And a little bit, that's the one. If you click there.
Click the share.
Click there to the question 1 abnormal ECG lab.
I'm not seeing it.
No, you're not there.
Oh, okay. I can see it now. I get what you're saying.
Click on there, yeah. And then you can click yes, you have completed that.
Oh no, so I can do it again.
You can do that as well. That's absolutely fine.
Yeah.
And then you work through that, and then you answer to those questions that it will present to you on that.
Okay.
That makes sense?
It does.
Perfect. And the same way you do for the other lab, it works exactly the same. What's important that you get the points is that you complete this part of the quiz where it is still within the connect. You need to do the lab, but you need to complete also this, what you're seeing now. So you get the full points.
Oh, so the one I did now is another point?
I think that it will add, but you want to make sure that you could do also this one.
Oh, okay.
Because if you don't complete this, you won't get full points.
Okay.
Make sense?
Yeah, it does.
Perfect. Do you have any other questions? Can I help with anything else? The other lab works like that.
No, I don't think I have a question.
Excellent. Well, I really appreciate you joining me tonight. I know that it was a lot of information.
It was.
But you did really well. And remember that this recording will be available for you. So you can go back later on and you can check anything that you kind of felt that went too fast and it'll be all set.
Okay.
Perfect. Well, I really appreciate you joining me today, and I hope you have very good rest of the night. If at any point you get questions later on, you just let me know, and I'll definitely make sure to check the messages often enough so you get an answer.
Thank you.
Thank you so much. I hope you have a very good rest of the night, and thank you for all the hard work you're putting to this course.
Thank you. Have a good night, too.
Have a good night. Bye. And we're just going to jump back to the PowerPoint just for a second. So I'll just remind us that any questions, you can always message me directly. I'll be so happy to help you. And this actually was very worthwhile, this last part, because it allowed us to have a look how to get these fields labs to work. So follow just the instructions that we just used to get the fields lab work through them. And if for some reason it doesn't work, I will have posted by the time this video uploads instructions in the announcement that what you should do then. There's an alternative activity, but please try to do the fields labs. It seems that they were still running quite nicely, so give it a go. All I have for today's session. I think that we've done pretty well. There was a lot of material, I know, to cover. And I do apologize that last night we had the massive power cut that meant that we did lose all of the power and internet, so we were not able to finish off. So this revision session was moved for today, and I will be uploading this video available for you. So that's all from me for today. Like I said, I appreciate all of you for all your hard work. I see really good work being done. I see really good grades being populated, and you can look forward to having your exam and practical grade released likely at the end of this week. So keep up the good work, and I will see you next week. Thank you.